I'd like to do a quick review of genetics because understanding how to do a simple genetics problem is very important to understanding inbreeding. And so let's take the usual case where if an individual is normal for a trait, they contain the dominant allele, but an individual that is phenotypically normal, that appears normal, can carry a recessive allele for the genetic disease, and they can pass that allele to their offsprings. So let's say we're going to cross two individuals that are phenotypically normal. That means they appear normal, but they carry the disease. And so at the top, I'm showing the cross. We're crossing a heterozygous normal individual with another heterozygous normal individual. Okay. And remember, a heterozygote contains a dominant allele and a recessive allele. And so if the first individual is a male, the first heterozygous normal individual is a male, that means 50% of his sperm cells will have the dominant normal allele, and 50% of his sperm cells will have the recessive allele for the genetic disease. The same thing for the female. If the female is heterozygous, 50% of her eggs will contain the normal allele, and 50% of her eggs will carry the recessive allele for the genetic disease. And so we set up the Punnett square that shows the potential fertilizations. Across the top, we have two kinds of sperm, and down the side, we have two types of eggs. And so if an individual with a normal sperm cell, if that normal sperm cell fertilizes an egg that contains the normal allele, you're going to have two dominant normal alleles. The individual is homozygous dominant, and the individual is phenotypically normal. Okay. If the sperm with the recessive allele fertilizes an egg with the normal allele, you're going to have a heterozygote with a dominant normal allele, but that person will be a carrier. It will carry the genetic disease. That individual will be heterozygous, and that individual will be phenotypically normal. And so again, if you have a dominant allele in one of the gametes, one of the sex cells, and a recessive allele in the other sex cell, you're going to get a heterozygous individual that is phenotypically normal. But again, that individual carries the genetic disease. If a sperm cell with the recessive allele fertilizes the egg cell with the recessive allele, that individual will have two recessive alleles. They're homozygous recessive, and they will show the disease. And if it's a lethal disease for the baby will die of the disease. And so two individuals that are carriers have a 20% probability of producing a kid that will die of the disease. And so let's take a look at genetic diversity. Genetic diversity is extremely important to the long-term survival of a species. And there's all kinds of levels of genetic diversity. Okay. We can have genetic diversity at the chromosome level, like we just talked about, an individual that's heterozygous, that has a dominant and a recessive allele on a particular chromosome for a particular trait, or on a particular pair of chromosomes for a particular trait, would carry more genetic diversity than if you had two dominant alleles and that individual was homozygous dominant. So we have it at the individual gene level. Within chromosomes, we have it at the individual level of the organism. And then all the organisms that form a breeding group are a population, and so we can have genetic variation at the population level. And typically, the larger your population size, the greater the degree of genetic variation you have in your population. And so that takes us to the concept of minimum viable population size, or MVP. The minimum viable population size is the minimum number of individuals you need in your population to prevent problems with inbreeding. And most of the studies have been done with mammals. And in mammals, for short-term survival, and you need to know that short-term survival, I'm talking about 100 years or less, the minimum viable population size is 50 individuals. So you need at least 50 individuals in your population for them to be able to survive 100 years or less. If you want your population to survive greater than 100 years, the minimum viable population size is 500. You need 500 individuals in your population to be able to survive more than 100 years. And these are numbers I want you to know from memory, both MVP for short-term survival and MVP for long-term survival. So let's think about this. Let's think about why. If you have a small population, you have much greater potential to have inbreeding, okay? And inbreeding increases the probability of unfavorable traits being expressed, okay? And if you remember that genetics problem we went over, negative traits are usually recessive. Okay, so they would be masked by the dominant normal allele. The other thing is negative traits are rare in a population. And so even if an individual carries a negative trait, is heterozygous for that negative trait, since it's rare in the population, the probability that that individual would mate with an individual that also carries a genetic disease would be rare or unlikely, okay? Then remember, negative traits, you need both recessive alleles. Negative traits are usually expressed when the genotype is homozygous recessive. You have to get a recessive allele from one individual and a recessive allele from the other individual, okay? Negative traits are masked by dominant alleles. Since they're recessive, they're masked by the presence of a dominant allele, and they're not expressed in heterozygous individuals. However, negative traits run in families. Now, if I have a population that's very large, it's highly unlikely that I'm going to mate with a person that's closely related to me, okay? Or if and in fact, in human populations, culturally, it's taboo to make babies with your brother or sister or mother and father. And there's a reason for this, okay? And that is because negative traits run in families. So if I breed with someone unrelated to me and I carry the disease, it's highly unlikely that I will produce an individual that is homozygous recessive, that I would produce an individual that would express that genetic disease. But if that disease is in my family and I make babies with my brother or my father, it is highly more likely that they will carry that recessive disease, and it is highly more likely I will create a baby that is impacted by that genetic disease. Okay. And so again, negative traits are usually recessive. Negative traits are rare in a population, and so if you have a big population size, the probability of inbreeding decreases. But when you have small population sizes, since negative traits run in families, and with a small population size, the probability of breeding with someone closely related to you increases. The probability of producing a baby that has that genetic disease is going to increase significantly. Studies of bighorn sheep, again, minimum viable population sizes relate to problems with inbreeding. And you're going to actually read this read an article on this bighorn sheep study. But the summary of the article, if they had populations of 100 or more individuals, they were able to persist up to 70 years. Populations with fewer than 50 individuals went extinct in less than 50 years, basically because of genetic deterioration, okay? Genetic deterioration related to inbreeding. Cheetahs in Africa are highly inbred. If you take a piece of skin, if you graft a piece of skin from one cheetah and graft it on another, there's no tissue rejection, which indicates they're genetically closely related. Think about that. If I need a kidney, and let's say for an A in this class, you will donate me your kidney, okay? And so they're going to use your kidney for a kidney transplant for me. You are so genetically dissimilar to me that it's highly unlikely that kidney transplant would not work. My body would see the proteins on your cells as foreign, and my body would attack that kidney that they put in me. And that is called tissue rejection. Okay. And so for organ transplants, they typically want to use someone closely related to you because there's much less likelihood that that organ will be rejected. Yet cheetahs in Africa are so genetically similar that you can take a piece of skin from one cheetah, graft it onto another cheetah, and there's no tissue rejection. Well, what are some of the problems with inbreeding in cheetahs in Africa? Extremely high juvenile mortality. Okay. Lots of babies born to cheetahs do not make it into adulthood due to genetic diseases. And also associated with inbreeding is low testosterone levels in males and actually abnormal sperm production. And so with inbreeding in cheetahs in Africa, you have high juvenile mortality. You have low testosterone levels in males, which reduce the probabilities that cheetahs will mate. And then you also have abnormal sperm production. There's a population of lions that were isolated in Ngorongoro crater in Tanzania. The population number crashed, and that population of lions on Ngorongoro crater still has problems with inbreeding. And these lions experience the same problems that the cheetah population in Africa experiences. High juvenile mortality, low testosterone levels in males, and abnormal sperm production. And so we covered genetics because I wanted you to understand some of the terminology associated with it, because genetic variability is extremely important to the long-term survival of wildlife populations. Genetic variability gives them resistance to disease, and it also gives them an ability to respond to environmental change. And in fact, a buzzword associated with genetic variability is hybrid vigor. And so if a population is said to have a high degree of hybrid vigor, that means there's a high degree of genetic variability in that population. So in summary, inbreeding tends to fix undesirable traits. High juvenile mortality is associated with inbreeding. And inbreeding results in the loss of genetic variation or a loss of heterozygosity. And therefore, inbreeding reduces a population's chance of surviving environmental change.